Okay, let's talk a bit about urine formation. Well, last time we talked about the glomerulus and the proximal tubule. Let's focus a bit on the loop of Henle, on the distal tubule, and the collecting duct. But let's do a small recap too. So tell us a bit about the sodium potassium ATPase in the proximal tubule, but please, instead of saying Na and K, say sodium and potassium because you get it confused when you speak.
All right, let's dive into urine formation, starting with a quick recap of the sodium-potassium ATPase in the proximal tubule, then focusing on the loop of Henle, distal tubule, and collecting duct, keeping it clear and saying sodium and potassium. In the proximal tubule, the sodium-potassium ATPase is a pump on the basolateral membrane of tubular cells, actively transporting 3 sodium ions out into the blood and 2 potassium ions into the cell, using ATP. This sets up a low sodium gradient inside the cell, driving passive reabsorption of most filtered sodium, water, glucose, and other solutes from the tubule lumen, concentrating the filtrate. Now, the loop of Henle, a U-shaped structure, creates a concentration gradient in the kidney medulla to conserve water. Its descending limb is permeable to water, which leaves the filtrate as the surrounding medulla gets saltier, concentrating the urine. The ascending limb is water impermeable, but actively pumps sodium and chloride out, diluting the filtrate and making the medulla hypertonic. The distal tubule fine-tunes electronic balance, reabsorbing sodium and calcium under aldosterone's influence, which boosts sodium-potassium ATPase activity, and secreting potassium and hydrogen ions to regulate blood pH. The collecting duct, the final stop, adjusts urine concentration based on antidiuretic hormone, ADH. When ADH is present, it becomes water permeable, reabsorbing water to concentrate urine. Without ADH, dilute urine is excreted. In short, proximal tubule's sodium-potassium ATPase drives bulk reabsorption. Loop of Henle sets up a medullary gradient, distal tubule tweaks electronics, and collecting duct controls water under hormonal control. Want more on hormonal regulation or specific disorders?
Why don't we talk a bit about the loop of Henle then? So, we've got a descending and an ascending limb, and we have some extraction of water in the descending limb, and an ascending limb we have extraction of Na and Cl. And I wonder why it's not the other way around. Yes. Tell us what juxtamedullary nephron and cortical nephron means too.
All right, let's zoom in on the loop of Henle, explain why water is reabsorbed in the descending limb and sodium and chloride in the ascending limb, and clarify juxtamedullary and cortical nephrons. The loop of Henle's descending limb is permeable to water but not solutes, so as it dips into the increasingly salty kidney medulla created by the ascending limb, water passively leaves the filtrate, concentrating the urine. The ascending limb, conversely, is impermeable to water, but actively pumps sodium and chloride out into the medulla via transporters like the sodium-potassium chloride cotransporter, diluting the filtrate and making the medulla hypertonic. This setup isn't reversed because it creates a countercurrent multiplying system. The descending limb's water loss concentrates solutes, which the ascending limb's sodium and chloride reabsorption amplifies, building a steep medullary gradient, up to 1200 milliosm per osma, that allows the collecting duct to reabsorb water and concentrate urine efficiently. If it were the other way around, the gradient would collapse, and the kidney couldn't conserve water effectively. Now, nephron types. Juxtamedullary nephrons, about 15% of total nephrons, have long loops of Henle that extend deep into the medulla,
So
maximizing
tell us again why it would collapse. I don't understanding. And why we have no active transport of ions. But instead it's these like aquaporine channels. And yeah, some passive movements via tight junctions.
All right, let's clarify why reversing the loop of Henle's function would collapse the concentration gradient, and explain the role of aquaporin channels, lack of active ion transport in the descending limb, and passive movements via tight junctions. The loop of Henle creates a medullary concentration gradient, hypertonic medulla for urine concentration. The descending limb is water permeable via aquaporin channels, allowing water to passively exit into the salty medulla, concentrating the filtrate. The ascending limb actively pumps sodium and chloride out, using sodium-potassium chloride cotransporters, making the medulla saltier and diluting the filtrate. If reversed, say, sodium and chloride were pumped out in the descending limb and water reabsorbed in the ascending limb, the gradient would flatten. Pumping ions out early would dilute the medulla, and water reabsorption later wouldn't build the hypertonic environment needed for the collecting duct to concentrate urine, wrecking water conservation. The descending limb lacks active ion transport because its job is to concentrate filtrate passively. Aquaporins, water channels, let water follow the medulla's osmotic gradient, no energy required. Tight junctions in the descending limb are relatively leaky, allowing minimal passive ion diffusion, but the ascending limb's tight junctions are tighter, preventing water leakage while sodium and chloride are actively or passively via channels reabsorbed. This countercurrent system ensures a steep gradient. Want more on aquaporins or how this ties to urine concentration?
I want you to tell me about sodium, and I remember, say the full name of the elements. Tell me about the sodium potassium chloride NKCC2 cotransporters, how they relate to the loop of Henle.
All right, let's dive into the sodium potassium chloride cotransporter and the KCC2 cotransporter, focusing on their roles in the loop of Henle, and I'll use the full names, sodium, potassium, and chloride, as requested. In the loop of Henle's thick ascending limb, the sodium potassium chloride cotransporter, NKCC2 isoform, is a key player. It actively transports one sodium ion, one potassium ion, and two chloride ions from the filtrate into the tubular cells, powered indirectly by the sodium-potassium ATPase, which keeps intracellular sodium low. This reabsorbs about 25% of filtered sodium and chloride, diluting the filtrate and pumping these ions into the medullary interstitium, building the hypertonic gradient crucial for water reabsorption in the collecting duct. The potassium brought in mostly recycles back to the lumen via potassium channels, maintaining the transporter's activity. This process explains why diuretics like furosemide, which block NKCC2, cause dilute urine and disrupt the gradient. The KCC2 cotransporter, potassium chloride cotransporter 2, however, is primarily found in neurons, not the kidney, and isn't directly involved in the loop of Henle. You might be thinking of the KCC family, like KCC3 or KCC4, which can appear in renal cells but have minimal roles in the ascending limb compared to NKCC2. If KCCs are present, they passively move potassium and chloride of cells to regulate cell volume, not drive the loop's main ion reabsorption. In short, sodium potassium chloride cotransporter, NKCC2, in the thick ascending limb, reabsorbs sodium, potassium, and chloride, creating the medullary gradient. KCC2 is neuron-specific and not relevant here. Want clarification on other kidney cotransporters or how NKCC2 affects urine formation?
I want to ask you about so we've got countercurrent multiplication, and I want to understand what that is.
All right, so we were just touching on the loop of Henle's role in urine formation, and you want to understand countercurrent multiplication. Countercurrent multiplication is the process in the loop of Henle that creates a steep concentration gradient in the kidney medulla, allowing the kidney to produce concentrated urine. The descending limb, Permeable to water via aquaporins, lets water exit into the increasingly salty medulla, concentrating the filtrate. The ascending limb, impermeable to water, actively pumps sodium and chloride out via the sodium-potassium chloride cotransporter, diluting the filtrate and making the medulla saltier. Because the loop's limbs run in opposite directions, counter-current, this process multiplies the medullary osmolarity from 300 milliosm per osma at the cortex to 1200 milliosma deep in the medulla. This gradient lets the collecting duct reabsorb water under ADH control, concentrating urine to conserve water. Without countercurrent flow, the gradient would be weak and urine would stay dilute. Want me to explain how this ties to ADH or specific disorders?
I wanna ask you about the distal tubule now. What's going on with the distal tubule? Tell us about the sodium Cl cotransporter and the renal outer medullary potassium channels.
All right, let's focus on the distal tubule and what's happening there, specifically with the sodium chloride cotransporter and renal outer medullary potassium channels. The distal tubule, following the loop of Henle, fine-tunes urine composition, adjusting electrolytes and acid-base balance. The sodium chloride cotransporter, NCC, in the distal convoluted tubule actively reabsorbs sodium and chloride ions from the filtrate into the tubular cells, driven by the sodium gradient created by the sodium-potassium ATPase. This reabsorption, regulated by aldosterone, helps control blood pressure and volume by retaining sodium, while chloride follows to maintain electroneutrality. Thiazide diuretics target this transporter to increase sodium excretion. The renal outer medullary potassium channels, ROMK, also in the distal tubule, secrete potassium into the lumen, balancing potassium levels. These channels are active when aldosterone boosts sodium reabsorption via NCC or sodium channels, as the influx of sodium creates a lumen-negative potential that drives potassium out through ROMK. This potassium secretion is critical for preventing hyperkalemia and is influenced by potassium intake and acid-base status. In short, the distal tubule's sodium chloride cotransporter reabsorbs sodium and chloride for volume control, while ROMK channels secrete potassium to regulate levels. Both
And what are principal cells?
Principal cells are specialized cells in the late distal tubule and collecting duct that regulate sodium, potassium, and water balance. They reabsorb sodium through epithelial sodium channels, ENEC, stimulated by aldosterone, which creates a lumen-negative potential, driving potassium secretion via renal outer medullary potassium channels, ROMK. They also express aquaporin 2 channels, controlled by antidiuretic hormone ADH, to reabsorb water, concentrating urine. Their role is critical for blood pressure, potassium homeostasis, and urine concentration. Want to dive into their hormonal regulation or related disorders?
Yeah, I want to understand principal cells a bit more.
All right, let's dive deeper into principal cells in the late distal tubule and collecting duct. These cells are key players in fine-tuning electrolyte and water balance. They have epithelial sodium channels, ENEC, on their luminal membrane, which reabsorbs sodium from the filtrate, driven by the sodium-potassium ATPase on the basolateral side that pumps sodium into the blood. Aldosterone ramps up ENEC activity, increasing sodium reabsorption to raise blood volume and pressure. This sodium influx makes the tubule lumen more negative, pushing potassium out through renal outer medullary potassium channels, ROMK, into the urine, regulating blood potassium levels. Principal cells also have aquaporin 2 channels, inserted in response to antidiuretic hormone, ADH, allowing water reabsorption to concentrate urine and maintain body fluid balance. Their strategic location and hormone sensitivity, aldosterone for sodium, potassium, ADH for water, make them crucial for homeostasis. For example, in hyperaldosteronism, excessive sodium retention and potassium loss via these cells can cause hypertension and hypokalemia. Want me to
Perfect.
explore?
Since you brought this up, explain to me now how create a nice system with critical thinking. Connect me the renin angiotensin aldosterone system with the principal cells and in the distal tubule, and 2-3 sentences, nothing crazy.
The renin-angiotensin-aldosterone system, RASS, regulates blood pressure by triggering renin release from the kidneys when blood volume or pressure drops, leading to angiotensin II production, which stimulates aldosterone secretion from the adrenal glands. In the distal tubule and collecting duct, aldosterone acts on principal cells, boosting epithelial sodium channel, ENEC, activity to reabsorb sodium, increasing blood volume, and enhancing potassium secretion via ROMK channels. This coordinated system ensures fluid and electrolyte balance, with principal cells as the key effectors linking RASS to tubular function. Want to dive into RAS disorders or specific triggers?
Well, I want to ask you about the collecting duct now and the difference between intercalated cells and principal cells.
All right, let's focus on the collecting duct and the differences between intercalated cells and principal cells. The collecting duct is the final segment of the nephron, where urine concentration and acid-base balance are fine-tuned. Principal cells, as we discussed, handle sodium reabsorption via epithelial sodium channels, ENEC, under aldosterone control, secrete potassium through ROMK channels, and reabsorb water via aquaporin II channels when antidiuretic hormone, ADH, is present, concentrating urine. Intercalated cells, found alongside principal cells in the collecting duct, focus on acid-base regulation. Type A intercalated cells secrete hydrogen ions via H plus ATPase to correct acidosis and reabsorb bicarbonate, while type B intercalated cells secrete bicarbonate to correct alkalosis. Unlike principal cells, intercalated cells don't manage sodium, potassium, or water balance, but are critical for pH homeostasis. In short, principal cells regulate sodium, potassium, and water for volume and electrolyte balance. Intercalated cells control acid-base balance by tweaking hydrogen and bicarbonate. Want more on how these cells interact or their clinical relevance?
Interesting.
Grazie.
Okay.
Vuoi approfondire le cellule
Look,
intercalated?
every time you hear me and you think I'm speaking in another language, I just want you to know I'm not. So instead of answering in another language, say, you got me, and then speak in English. Okay, let's continue. So let's talk a bit more about ADH and the collecting duct now, and V2 receptors and aquaporin 2 channels.
You got me. All right, let's dive into ADH, antidiuretic hormone, the collecting duct, its 2 receptors, and aquaporin 2 channels. ADH, also called vasopressin, regulates water reabsorption in the collecting duct to control urine concentration and body fluid balance. It acts primarily on principal cells through 2 G protein coupled receptors, V1 and V2. The V2 receptor, most relevant here, is on the basolateral membrane of principal cells. When ADH binds V2, it triggers a signaling cascade that inserts aquaporin 2 water channels into the luminal membrane, allowing water to move from the dilute filtrate into the hypertonic medulla, thanks to the loop of Henle's gradient, concentrating the urine. Without ADH, aquaporin 2 channels are removed, producing dilute urine. The V1 receptor, less prominent in the collecting duct, is more involved in vascular smooth muscle, causing vasoconstriction to raise blood pressure. In short, ADH via V2 receptors in the collecting duct's principal cells boosts aquaporin 2 insertion, enabling water reabsorption for concentrated urine. V1 handles blood pressure elsewhere. Want to explore ADH disorders like diabetes insipidus or how this ties to blood volume?
I want to talk a bit more about this because I really don't understand these vasopressin 2 receptors.
All right, let's zoom in on the vasopressin V2 receptors in the collecting duct to clear things up. Vasopressin, or antidiuretic hormone ADH, controls water reabsorption in the kidney's collecting duct via V2 receptors, which are G protein coupled receptors found on the basolateral, blood facing membrane of principal cells. When ADH binds to V2 receptors, it activates a signaling pathway involving cyclic AMP, CAMP, and protein kinase A, which prompts vesicles containing aquaporin 2 water channels to fuse with the luminal, urine-facing membrane. This allows water to pass from the dilute filtrate in the collecting duct into the hypertonic medulla, concentrating the urine and conserving water for the body. Without ADH, V2 receptors stay inactive, aquaporin 2 channels are internalized, and the collecting duct remains water impermeable, producing dilute urine.
Okay, I want to ask about diabetes insipidus. Explain to us what insipidus means. And explain to me nephrogenic and neurogenic too. Talk to me about treatments too, like chlortalidone, desmopressin, vasopressin, and yeah. I would also like to know about the syndrome of inappropriate ADH.
All right, let's dive into diabetes insipidus. Break down what insipidus means, explain nephrogenic and urogenic types, cover treatments like chlorothalidone and desmopressin, and touch on the syndrome of inappropriate ADH, SIADH. Diabetes insipidus, DI, is a condition where the kidneys can't conserve water, leading to excessive dilute urine and thirst. The term insipidus, from Latin, means tasteless, as opposed to diabetes mellitus, sweet urine, highlighting the lack of glucose in DI urine. There are two main types: neurogenic or central DI, caused by insufficient ADH production in the hypothalamus or pituitary, due to trauma, tumors, or genetics, and nephrogenic DI, where the kidney's V2 receptors in the collecting duct don't respond to ADH from genetic mutations, drugs like lithium, or kidney disease. In neurogenic DI, the body can't signal water reabsorption. In nephrogenic DI, the signal is ignored, both resulting in dilute urine and dehydration. Treatments vary by type. For neurogenic DI, desmopressin, a synthetic ADH analog, is given nasally, orally, or by injection to mimic ADH, binding V2 receptors to insert aquaporin 2 channels and concentrate urine. For nephrogenic DI, desmopressin is less effective since V2 receptors are defective, so treatments focus on managing symptoms. Chlorothalidone, a thiazide diuretic, paradoxically reduces urine output by increasing sodium reabsorption in the distal tubule, concentrating the filtrate upstream and boosting water reabsorption in the collecting duct, even without ADH. Other approaches include low-sodium diets or drugs like amiloride for lithium-induced cases to improve kidney response. The syndrome of inappropriate ADH, SIADH, is the opposite of DI, where excessive ADH secretion from tumors, lung disease, or medications causes too much water reabsorption, leading to concentrated urine, low blood sodium, hyponatremia, and fluid overload.
Okay, how can nicotine, morphine, and alcohol affect diuretic action?
All right, let's dive into how nicotine, morphine, and alcohol affect diuretic action, tying it to urine formation. Nicotine, a stimulant, can indirectly reduce diuretic action by stimulating antidiuretic hormone ADH release from the pituitary, likely via stress or sympathetic activation. Increased ADH enhances aquaporin 2 channel insertion in the collecting duct, boosting water reabsorption and concentrating urine, counteracting diuretics. Morphine, an opioid, also suppresses diuretic action by stimulating ADH release, often
Interesting.
through.
Well, thank you for watching, guys. I'll see you at the next episode. Peace.